Good afternoon, everyone. Barb is a difficult show to follow because she presents such an upbeat kind of presentation. I'm sorry to say mine is not going to be that upbeat, okay? It's probably the opposite. So I didn't really plan to say anything about myself, but I saw everyone else did, and because I thought, I just could look at the bios that we have in the booklet. And also, anything I would have to say probably is not very relevant to this group. But I'll briefly tell you that I'm actually an ion channel biophysicist. I did research, structure function research on channels for many, many years. Through a very interesting and long story, I became a program director at NINDS, again with a portfolio about ion channels and transporters. And that's where I discovered my mission, which is improving the quality of science, okay? So that's kind of what I'm going to be talking about. But why am I here? I actually have some imposter syndrome, okay? I've never done any translational research. I've actually never even done in vivo studies in animals, okay? And not only that, I'm presentation number 13. And if you think that's bad, 2 weeks ago, I was assigned a new parking spot at NINDS, and the number is 13. Okay, so yeah, I have some issues. So really, why am I here? And I think the reason I'm here is to be a Jiminy Cricket. Okay, I'm here to appeal to your conscience. Okay, and we all know that the conscience resides in the heart, not in the brain. Okay? So I hope to change your hearts. Okay, so that's really what I'm trying to achieve today, and you really just follow along and see what we can get. But before we start, I have a disclosure to make, and that is opinions I will voice are not official opinions of NIH. Okay, it's very important to say, and as you'll find out, I have lots of opinions. Okay. So you've heard a lot about the translational pipeline, and here you see the odds of success from moving from one phase, clinical phase, to the next. This is the latest study I could find that was pre-COVID, because COVID kind of shuffled things around. And as you can see, to go from phase 1 to phase 2, you have about 50% chance, okay, 1 and 2. Phase 2 to 3, it drops quite significantly, and then phase 3 to NDA, and then to approval. Now, if you calculate the probability of going from phase 1 to approval, that comes to 7.9%. Okay, I told you I'm not going to be upbeat. Okay, now if you think this is bad, in neurology it's only 5.9%. Okay, so the question that I pose is this a rule of nature? I mean, is this just what we have to deal with? Or can we do better, okay? And I'm a strong believer that we can do much better, okay? And that's really what we're going to be talking about. So in Mike's open presentation, when he just kind of kicked off this workshop, he divided the pipeline into 2 sections. He talked about preclinical, which he said is not regulated, and the clinical, which is regulated. Okay. Now it's important to note that the preclinical, of course, goes from the very basic scientific discoveries, right, to the point that you all want to go into or are already in, which is the translational phase, and hopefully, to get to the next step. Okay. Now, Mike is a really nice guy, so. He says not regulated, but not regulated is synonymous with unregulated, which doesn't sound so positive. And if you look at the definition of unregulated, you can see that what's highlighted is disorderly and chaotic, okay? Mike, as being a nice guy, says that's not too bad because, that's how you'll discover new things. I always look at the half-empty glass. So, to me, this says, oh my God, chaotic, disorder, that's not very good. However, that means there's room for improvement, okay? And if there's room for improvement, that means maybe we can improve the probability of success, okay? So coming back to the odds, I'll be focusing primarily on the phase leading up to where you are, okay? not kind of, during the translation, even though, it applies to that, and not into the clinical domain, okay? And I say, yes, we can improve the odds, and I'll focus on what I believe are contributing factors to the low probability of success. And that will be, I'll talk about the reasons for low success. I'll tell you what has been already done or being done to improve things, and what practices we should embrace. Okay? So I'll start by telling you a story, and it's a very sad story for two reasons. One, it's about ALS, which we all know is a devastating disease, right? And the other is the outcome of the story I'm going to tell you, which is very actually depressing, okay? And so, as you all know, and actually Guy already discussed the SOD1 mouse model. The SOD1 mouse model was discovered in the 1980s and was used extensively. It was the first animal model for ALS. It's been used extensively to look for treatments for ALS, okay? And in 2002, a couple of papers were published suggesting that minocycline can dramatically extend the lifespan of the SOD1 mouse, okay? Now, for various reasons that I can only guess because I wasn't yet at NINDS, within 1 year, NINDS funded a clinical trial to look at the effect of minocycline in humans, okay? And the reasons could have been many. I mean, it could have been the dramatic effect that minocycline had. It could have been that people were, obviously ALS is so devastating, and they their life expectancy is so low that we should try to find cures as quickly as possible, or the concern that minocycline is so easy to get hold of that if people just started taking minocycline under the assumption that it would be good, then there'll essentially be no patients that would be suitable for any other clinical trials because they'll be all on minocycline. And that also happened, by the way, with lithium many years later. So, as I said, in 2003, within 1 year, a clinical trial was initiated, 412 patients who were treated for 9 months. The results were published 4 years later, and it was found that minocycline had no beneficial effect. Okay? So why am I telling you this story? There's absolutely no reason to assume that what works in a mouse would necessarily work in humans. If that was the case, we wouldn't have to do human clinical trials. Okay? Well, the reason I'm telling you this story is because a year after the results of the clinical trial were published, a paper was published by the ALS Therapy Development Institute. It's a nonprofit in the Boston area. And they devoted 4 or 5 years to very rigorously test more than 70 compounds in the SOD1 mouse model. And they did these studies so carefully that they could actually detect, I'm running ahead of myself here, they actually could detect a statistically significant difference in the life expectancy between the female mice and the male mice on the order of about 3 days, okay? However, they found absolutely no effect for any of the 70 compounds, several of them who have been previously reported to be efficacious, one of which was minocycline, and several of which went on to clinical trials and also were not successful in clinical trials. Now, when I read this, I became incredibly depressed. And not only because of this outcome, and not only about the false expectations and hopes in these patients that obviously died shortly after these clinical trials, because that's their life expectancy, but also because the first author on this paper, Sean Scott, he had ALS and he died a year after this paper was published. And I could only imagine what he must have gone through knowing about all these false hopes, all these prior studies that led to nothing, because when you do things rigorously, the effects went away. Okay, the effects in the SOD1 mouse model. And by the way, that's why the background of my slides is black. Okay, so very depressing. Now, this study came out at a period where there were a flurry of publications, just a small fraction of which are shown here, suggesting that there's problems with the quality and transparency of preclinical studies. okay? And to give you just one example, it's one of my favorites because I'll explain in a moment why. But this is a paper that was published by Bayer Healthcare, and they reported that out of 67 projects that they started in the company, and when I say started, I mean they devoted significant effort to try to advance these studies that initiated from publications by the academic community, okay? In 2/3 of cases, they could not reproduce the results, okay? Now, the reason I find this so important, this paper, because to my knowledge, this was the first time that pharma openly said, hey guys, we've got a problem with academia, okay? I mean, up till that point, you know, in the hallways, you always heard, talk about these things. But this is the first open one, okay? Now, there could be many reasons why results cannot be reproduced. Some of them are very good. You know, if you're doing research on the cutting edge of science, you wouldn't expect necessarily that you'd be able to reproduce the results, right? Or there could be many other good reasons, okay? However, I think that part of the problem are 3 areas which we're going to discuss today. One is essentially lack of transparency. Okay? People don't report what they actually did, and therefore it's almost impossible to be able to reproduce what they did. Okay? The other is experimental bias, which I call human nature. And the third is chance and publication bias. Okay? And we'll go through all these 3 areas. And I want to start with human nature. Okay? 400 years ago, Francis Bacon, in his book, The New Organon, wrote, once a man's understanding has settled on something, it draws everything else also to support and agree with it. Okay? He went on to say that it is an innate and constant mistake in the human understanding to be much more moved and excited by affirmatives than by negatives. Okay? Now, what's he talking about? He's talking about unconscious biases. Okay? And we all know, and he did probably too, that understanding resides in the brain. And that's why I like the quote by the comedian Emma Phillips, who, a contemporary scientist, so to speak, who said, I used to think that that the brain was the most wonderful organ in my body, then I realized who was telling me this. Okay? So there's unconscious biases, okay? Now, there are many kinds of unconscious biases, and I just want to play this little game with you. And if I can see a show of hands, who's seen this figure before? Faculty, of course, raise their hands, yeah. But anyone else have seen this before? Who hasn't seen it? Okay, well, you're still awake, okay. So what I have here are 2 tables, and I labeled one side of each table with a W, okay, to indicate that that's the width of the table. And I'm going to ask you the most idiotic question you can imagine. My question is, which table is wider, okay? Now, if I can see a show of hands, who thinks the table on the left is wider than the table on the right? Okay. Sorry? How's your stage left? Left, the way, your left, not my left, okay? Okay, so for the record, no one raised their hands. Who thinks the table on the right is wider than the one on the left? For the record, there are many hands up. Who doesn't think? There's always one, okay? Okay, so let's put this to the test. I'm going to cover the left table with a rectangle, and I'm gradually, very slowly going to move it over so you can see that there's no trickery here, okay? These tops are identical. And no matter how many times I'll show this to you, you will still think that the table on the right is wider than the table on the left. And I've done this hundreds of times, and I still see that, okay? And if you don't believe me, you can do what I did. I printed this out, cut out the top, and put them one on top of the other, and saw that they are identical, okay? So why am I giving you this example? Do we rarely care as scientists which table is wider? Not really, okay? But I want you to think how many of the things we do as scientists rely on our vision. if it's immunohistochemistry, okay, Western blots, functional MRIs, selecting cells for electrophysiology or for whatever, okay? Think how many things we assume that what we see is what we get. But our brain is playing tricks on us, okay? So that's something to keep in mind. Now, there are many kinds of bias, and because there's all these unconscious biases, how do we wrap our brain around it, so to speak, okay? And I love the definition that was put forward by David Ranzerhoff. And he said, the reliability of his study is determined by the investigators' choices about critical details of research, design, and conduct. What he's telling us is it's our responsibility, okay? It's our responsibility as scientists. And he goes on to say that bias is unintentional and unconscious. So if it's unintentional and unconscious, how can we be responsible? Okay? So he gives the solution for that. What he says is when you do an experiment, you've got to make sure that everything is equal between comparison groups during the design, conduct, and interpretation of the results. Okay? Everything equal except for that one parameter that you're checking. Okay? Let's look at an example. So this is a very interesting, subtle example. This was a study that looked at the effect of what's it called? What do they call it? Neuroserpin in the differentiation of neurons in culture. And I'm not an expert in differentiation of neurons in culture, but they kind of have arrows to suggest that the differentiation is different. Okay? A few months after this paper was published, they retracted it. Why did they retract it? Because they realized that they didn't make everything equal between comparison groups. So what went wrong? Neuroserpin was either expensive, in low supply, or for whatever reason, they had a little of it, and what they did is, when they fed the cells in culture with neuroserpin, they mixed it in media and filtered it through a small syringe and filter. Okay? Media is cheap, so the controls were the media was just filtered through, in bulk. Okay? After the paper was published, they either got more neuroserpin or, for whatever reason, they filtered it in bulk and the effect went away. Okay? Now, to their credit, they retracted the paper and then devoted 18 months to try to figure out what went wrong. And they discovered that there's something being leached by the syringe and filter that gives rise to the effect. It had nothing to do with neuroserpin, okay? And then they published the result. I take my hat off to these scientists. They did an honorable thing. They set the record straight. I've got a lot of respect for them, but I wouldn't want to be in their shoes. And even though I was a rigorous scientist since I was about 5 years old, I can imagine how many times I probably didn't make everything equal during comparisons. I could easily see how something like this could happen. This is a minor little thing, right? You'd have to think of so many things to make everything equal. And that's on us, okay? Okay, let's come back to our minocycline story. This is a story that I told you about, the study that came out and that led to the clinical trial, the couple of studies that led to the clinical trial. And we'll come back to the story periodically through this talk. And the first paper that described this was published in Nature, not surprisingly, okay? And the key figure is what you see here. It's a survival plot, and you can see the control animals in the dashed line, the minocycline-treated animals in the solid line. And when you're doing this kind of experiment, what is the very first thing you have to do to make sure that you're making your groups as equal as possible? What's the very first thing you will do? Anyone? Randomize, okay, right? You have to randomize the animals to control groups, right? Makes perfect sense, right? The Nature paper makes no mention of whether there was randomization or not. A second paper that came out that same year, also no mention of randomization. Okay? Let's look at another example of kind of bias that can creep in. And actually, this is a study that I found relatively recently. I always used to give an example from like 60 years ago, and this is a relatively recent one. And I particularly like this one because speaking about rigor and making everything equal between comparison groups, this is a fascinating study that they did, okay? So what did these investigators do? So this was an experiment they did with veterinary students, third-year veterinary students. And when you're a veterinarian, you don't sit in your office and patients come to you and with complaints, right? You've got to figure out what's going on, okay? So what you typically do is you have to observe the animals and try to see what's wrong with them and so on. So a lot of important things happen by just observing the animals. And there's something called social breeding values, okay? Which we won't get into, but the students were trained to identify particular behaviors in the animals so that they can be good veterinarians, okay? And the experiment involved 157 students, and they were told that there were pigs that were selected to have this quality of being, favorable breeding characteristics, and of course there were control animals that weren't. And so after being trained how to identify these things, they were shown a 5-minute video, okay? And in the video included a pen with 6 pigs, and they were supposed to identify positive and negative behaviors of these pigs, okay? And they were shown a second video of 5 minutes, where they were also supposed to do the same thing. One was the so-called positive breeded animals, and the other was the control, okay? And the students were divided into 2 groups. Half the group were told that the one on the left is the control, and the second half were told that the one on the right is the control. Now, the only difference between these 2 videos, it was exactly the same video. They just flipped it and changed the lighting a little bit to make it look like it's a different set of pigs, okay?
Pigs are the same size.
It's exactly the same. They're the same size. Yeah, you're right, yeah. Anne has a sense of humor. Okay. So that's good to see. Why did I say this is such an incredible experiment? Because if you think about it, they took half the students and made them think that the one is the control and half the student the other. So at the end of the day, they completely equalized the experiment. So they made everything equal between comparison groups. And what did they find? Okay, again, this is exactly the same videos. They found, as you would expect, because they were told that there would be advantages to one group versus the other, that they have more positive social behavior, and the ones that so-called were the SBV animals, and fewer negative behaviors. Okay? This is what's called expectation bias. Okay? And we all suffer from it. Okay? So I found this study extremely interesting. Okay, now it doesn't take a rocket scientist to figure out what could have been done to prevent this expectation bias, right? All that had to happen was not to tell them, which group is which. Just say, do you see a difference between the 2 groups, right? So there would be no expectation. Now, the way you do that is obviously by blinding them, right? Now, when I tell people, you should blind your experiments, a lot of times it's a little scary, okay? I get all these reactions. Oh, we can't do that. Oh my God. I need more money. I need a bigger lab. All these kind of. But in reality, I'm looking at pink mice versus green mice. How can I possibly do it? So in reality, though, in many cases, it's not that difficult. All you have to do is ask someone in your own lab or in the adjacent lab to code the samples and not to reveal the code until the experiment has been conducted and analyzed, okay? So really, in many cases, you can do that. And there's an advantage of going to ask someone else to do this because you never know what might come out of it, okay? Okay, let's go back to our minocycline story, okay? The Nature paper makes no mention of blinding. The other paper says the experimenter was blinded to the treatment protocol. And I don't know what that means because you need to blind yourself at all stages of the experiment, okay? From animal selection to the treatment to the analysis, okay? So I don't know what they actually did there. Now, all I've told you so far is that a couple of groups of investigators who studied the effects of minocycline didn't report whether they randomized or blinded, okay? That's all I told you. So how widespread is this phenomenon? So let's look at an analysis that was done. They looked here at 4 neurological disorders, this group, and we'll focus just on one, on multiple sclerosis, because it had the largest number of papers that they looked at. They looked at 1,117 papers to see what is reported and what is not, okay? And they found that only 16% reported on blinding of the assessment of outcome, okay, of the analysis. Only 9% reported on randomization, and look at this number at the end. Only 2%, only 2 papers, okay, out of 1,117 studies wrote in the paper how they came up with the sample size. Okay? It's pretty, outrageous, right? Okay? That wouldn't happen in a clinical trial, obviously. Okay? So, this just tells us that they're not in the habit of reporting, but do we know if they actually did it or not? And that's more difficult, obviously, to tell. If you pick up the phone and say, hey, did you do that? What answer are you going to get, okay? So it's going to be difficult to do it that way. But there's indirect evidence that actually lack of reporting is associated with lack of practice. And this is a really cool study. They looked at all the papers that reported the effect of the immunosuppressive drug FK506 in experimental stroke. And for each paper, they gave a quality score. They said, like, did they report on blinding? They got a point. Did they report on randomization? They got a point. Did they provide information on sample size? They got a point. And they could accumulate up to 10 points, and that's the quality score shown on the x-axis. The y-axis shows the efficacy of FK506 as it's reported in each one of these studies. Okay? And I think it looks quite obvious that there's an inverse relationship. Okay? The fewer parameters that are reported, the bigger the apparent efficacy of the drug, suggesting that lack of reporting is associated with lack of practice, and that lack of practice leads to unconscious biases. Okay? Now, the examples I gave up till now are all in neuroscience, and it's no coincidence because actually neuroscience is leading in this effort to address issues of lack of transparency and rigor, but it's not unique to neuroscience. And in 2006, Hakam and Riedermeyer published this paper, which I find fascinating. What they did is they looked at 7 obscure journals that are listed there on the left to see if they could identify papers that had a high impact. And they defined high impact as papers that were cited at least 500 times each, that were cited in applications for doing clinical trial. They contributed essentially to approval of a clinical trial. And they all had to have, obviously, animal model studies in them, okay? And what they found is that only 20% of the papers that they identified reported on blinding, only 14% reported on randomization. It's pretty depressing. And when I read this paper, 2 questions came to my mind. One is, how could the reviewers who reviewed these papers decide that they are worthy of publication in one of these obscure journals without knowing if measures were taken to minimize unconscious bias? And the second question is, how could all those who cited these papers decide that they are worthy of citation without having that information? And there are many possible reasons. I'll give you 3 of my favorite. And I'll start with a cynical one, and uncharacteristically, and that is the definition of peer review in Wikipedia. Okay, Peer review is the evaluation of work by one or more people of similar competence to the producer of the work. Okay? Our reviewers are no better than us. They are our peers. If the entire community is not in the habit of randomizing and blinding and calculating sample sizes and et cetera, there's no reason to think that the reviewers would point this out. Okay? So that's one thing. The other is, I've here showing you the number of papers in PubMed in the English language published per year in the last 65 years. I mean, since 1956. Okay? And as you can see, the number of papers per year went up more than 30-fold. What does that mean? It means that scientists who are doing science today, as opposed to those who did it 65 years ago, have to read 30-fold more papers per year, to read 30-fold faster than 65 years ago, or 30-fold more superficially. Okay? So it's not surprising to me that people cite papers without really digging into them and looking carefully. Okay? And as an anecdote, when I was a student, there was a story that went around that Sir Bernard Katz, the very famous scientist, embedded in one of his papers a fictional reference. And then he looked to see who in the field will cite that reference because it didn't exist. Okay? Now, they may have said that just to educate me what not to do, but I think it's a cool idea. Okay, the third thing, and perhaps the most important thing, is this vicious cycle, which I call the perversive incentive of paper money. If you don't produce papers, you don't get the money. Okay? And we all know that the engine that drives our research is grant funding, and the fuel is the papers that will feed that engine. Okay? So we have to publish many papers, preferentially in high-profile journals, which will require that it'll be innovative and novel and highly significant and so on. And we basically have to feed this engine, on average, at least in this country, every 4 years. Okay? Now that's not realistic. I mean, it's just not realistic. How could we possibly expect, your garden variety investigator to do that? Okay? So what happens? Investigators rush studies to publication. Studies that could have benefited from a couple more controls. Studies that could have perhaps designed experiments to disprove the hypothesis, not to prove it right. Perhaps using larger sample sizes. Okay? They don't have the time. Okay? They're under tremendous pressure. So that's the problem with this. And the other thing that's generated by this vicious cycle is publication bias. And publication bias, I like the definition that was put forward by Kay Dickerson. She said publication bias is the tendency on the part of investigators, reviewers, and editors to submit or accept manuscript for publication based on the direction of strengths of the study findings. Now, the reason I like this so much is because she's actually pointing a finger at who's responsible for it. Okay? The other parties as well. But the investigators, the funders, the editors, okay? And there's evidence, by the way, for all of these. Okay? So, how widespread is this publication bias? There are many models that have been developed to try to figure out how much, what the extent of publication bias is. These are all built on assumptions because they're models, and I'm not a big fan of models where you have to make assumptions. I like good empirical data, okay? So here's an example of an empirical data. In this study, the investigators looked at 525 unique papers in experimental stroke. They found that only 10 of them, okay, 2%, did not report any significant effect on infarct volume. When I read this, I said to myself, wow, aren't we an amazing species? When we come up with an hypothesis, we're right 98% of the time. Okay? Now, obviously, that's nonsense, right? And obviously, we have a big problem with publication bias. And if you combine publication bias with the element of chance, you've got a perfect recipe for poor reproducibility. And to make this point, we're going to go back to our minocycline story for the last time. And in the paper that came out by the ALS Therapy Development Institute, the ones that did 70 more or so compounds, okay, they wrote in the abstract, the majority of published effects are most likely measurements of noise in the distribution of survival means as opposed to actual drug effect. Now, English is not my mother tongue, and it took me forever to try to understand what they were trying to say here. I read this over and over and over. And then I understood what they were saying. They were just being polite. What they were saying is that the results were by chance, okay? Now, in order to say that audacious statement, they had to have good evidence, and they had outstanding evidence. Because they looked at so many drugs in so many studies, they had 2,241 SOD1 mice that served as controls. 2,241. And they knew everything about them. They knew when they were born and when they died, what their sex was, what litter they came from, how many copies of the transgene of the SOD1 they had, and what their cause of death was. And they put all this information into a database, and then they ran this in silico experiment. They randomly selected animals from this database and put them in two separate groups, and they said, what is the probability of finding a significant difference in the life expectancy between these two groups that by definition would be by chance, because these are all control animals, right? And they defined significant difference as a 5% difference. Okay? So they could do this thousands of times because it's in a silico experiment, so you could see kind of really tight error bars. And if they had 4 animals per group, they had a 30% chance of seeing a significant difference. Okay? Now, you might say, who in their right mind would use only 4 animals per group? That's ridiculous, right? Well, do you remember those 7 obscure journals that we talked about? Half of the papers that were listed in that study used 5 or less animals per group. Okay? Now, if they went up to 10 animals per group, they now had a 1 in 10 chance of seeing chance observation, okay? And in the paper, they concluded that if you want to do things carefully, when you're looking at survival, I'm stressing here, when you're looking at survival in the SOD1 mouse model, if this doesn't apply to every kind of experiment you do, okay, then you should have 12 animals of each sex per group, okay? That's their conclusion. So let's come back to the minocycline story. If we look at the Nature paper, they used 10 animals per group. No mention of the sex, and we know that there's a difference in the life expectancy between sexes. So for all we know, they didn't adjust for that. The second paper had 7 females per group. So if we take everything together that I showed you about these studies of minocycline that led to a clinical trial, I think it would be fair to say that they found these effects either due to chance or due to unconscious biases. Okay? Yes?
The statistics that were provided in those graphs, they must have given some sort of p-value or some hazard ratio or something like that.
Could be. I don't know. I have a whole talk I give about the p-value pretense. Okay? So let's not go there. Do you really need, I mean, if we go back, you mean in the minocycline studies? Or in here? I mean, you just look at those error bars, I mean, it's minuscule, right?
Well, right, so if you have P005, there is, you can still, that means you have 5%.
Yeah,
yeah.
Okay. We can have a lot of interesting conversations later, after your talk. Okay, so with that, I'm done with talking about the minocycline story, and the question now is, okay, what can we do? Okay, how can we improve rigor? So the first thing we need to do is we need to improve transparency. Okay? And the way to achieve that is to address the review of manuscripts and grants. So in 2012, we held a workshop with editors of prominent journals, And that workshop led to a paper. It's the Landis et al., published in 2012. And it had a large impact on journals. And I know that, first of all, from the editors themselves, but also, the proof is in the pudding. And that is 8 months after the workshop, Nature Publishing Group made significant changes to their how they evaluate manuscripts. So what changes did they make? The first thing they did is they removed the restrictions on the length of the methods. Really? How idiotic was the restriction in the first place, right? The most important part of the paper is the methods, and that was restricted. So no surprise that there's not transparency. Okay? The other thing they did is they added an extensive checklist where you're required to provide all kinds of information. Did you blind? Where's it written in the paper? Did you randomize? Where's it written in the paper? How did you calculate your sample size? Where's that? This, a whole bunch of things. Okay, did you, did you validate your antibodies? Where does it, which ones, what, etc. Okay. People in the community knew that somehow I'm responsible for this. For the longest time, I had to wear a bulletproof vest and a helmet. It was not easy. Okay, other journals followed. We saw just in the previous, one of the previous talks, Science Translational Medicine. They were the next one to adopt this, and there were several other journals that did the same. What about grant review? NIH also made changes. They didn't call it changes; they called them enhancement to peer review. That's the political way of saying things. And they did. I'm showing this first came out in 2016. It was updated in 2019. I'm showing you the 2019 version. And it has 4 pillars. One is the rigor of the key supporting data, okay? And I cannot stress enough how important this is, okay? And particularly for you all. Not particularly. Also for you all, okay? You're about to, or already in, this adventure of trying to take things to the clinic and hoping to help people and so on. Is what you're doing based on solid data, or is it built on a house of cards? Okay? Have you really looked at this in an unbiased way? Okay? So that's incredibly important. And that's true when applying for a grant. You're asking for money to do something. What's it based on? Is it based on solid foundation or not? Okay, so I think this is perhaps the most important thing in grant review. To tell you that it's actually working, I can't say that, because who are our reviewers? Our peers, okay? So it's very difficult to change minds. This is going to take probably years. The other thing that they incorporated is you have to say what rigor criteria you're going to use in the studies you're proposing to do, okay? How are you going to minimize unconscious biases? Now, this is, promises, right? Because this is what you're going to do, okay? So that's why I think the first one is infinitely more important. The third thing they added was this biological variables, such as sex. I don't think that has to do with rigor. That has to do with maybe external validity, but not necessarily with rigor. And the fourth item was authentication of resources. And just to put things in perspective, some estimates place the number of cell lines that are misidentified at 30%. Okay, 30%. So you might be thinking that you're working on one cell line and you're actually working on something completely different. Okay, and we've already heard about how fantastic antibodies are. Okay, so how are you going to authenticate that the antibodies are really, what you say they are? Okay. Second thing is how do we improve actually how scientists do their experiments, and that has to be through I think educational and awareness. Awareness that's why I'm here, okay, and that's why I'm happy to go give talks everywhere, anywhere, because that's the way to get to the heart of people, and to actually maybe change behavior. Education. education is a major issue. I'm going to ask for a show of hands. How many here in this room had training in the principles of rigorous research? And I'm not saying experimental design, which is a course in statistics. Principles of rigorous research. Amazing. Very, normally it's a 0 or 1 foreigner, who trained overseas, okay? There's almost no training in the principles of rigorous research in this country, okay? And there are many good reasons why that can be. So we decided at NINDS to do something about it. And what we're doing is we're in the process of developing a platform for different components of the principles of rigorous research that'll be freely available to everyone in the community. It'll be harmonized. It will be modular, adaptable, upgradable, etc. And the idea behind it is that it can be used either as the foundation to build courses at the institutions, you can use it at the bench, if you're just sitting there doing an experiment and say, wait a minute, there was this guy who told me something I need to blind. I don't remember how to do that, you could go and look that up. Or in lab groups, when you're discussing various aspects of rigor in journal clubs or in designing experiments, etc. Okay? So this just kicked off August 1st of last year, so hopefully at the end of 5 years we'll have a very powerful educational platform that we all can benefit from. Finally, perverse incentives. How do we deal with that? Well, for that we need to change the culture. And good luck, right? Changing the culture. I mean, that's incredibly difficult. So we're doing our best what we can. We held a workshop this past May. We set it up in such a way that it was community-led, because it can't come from the government. So we had 2 co-chairs, Veronique and Steve, and we brought together people who are involved in trying to change the culture, the rigor culture. We call them rigor champions, and to see how we can galvanize the community, okay? And it was really fascinating workshop. Where this will lead to is still not clear, because the paper has not come out yet, so we don't know how the community will buy into this. We're doing other things. We have like a Slack channel for rigor champions. We now have a Twitter account, and doing the best we can. If you want to listen to a fascinating talk, so here you can see the link to the presentations of this workshop. The keynote was given by Damien, why am I blanking? Damien Centola, who spoke about how you get behavioral change. Fascinating talk. He also has a famous book by the name of Change, okay? So Damien Centola was the keynote here. Okay, so what's the take-home message? Right? The take-home message is we're all prone to bias. Right? That's nothing we can do about it. Okay? That's how our brain is structured. And therefore, we must critically assess results in publications. We must rigorously design and execute and analyze experiments. Okay? Plan experiments to disprove the hypothesis. Practice humility. Okay? Here we show that we have discovered, all those things, that's not humility. Okay? So practice humility when you're looking at your results and your experiments, and ask yourself, what did I do wrong? Where did I go? What did I do wrong? How can I make sure that what I'm doing is the right thing? And last but not least, you should favor large effects. Now, I said absolutely nothing about that, right? And I didn't, because we don't have time. So I'll leave you with one slide to get this point across. The odds of winning the lottery are 1 in 300 million and change, okay? Yet many people believe that they can win the lottery, and some people do. Okay? The odds of being struck by lightning in the United States in a given year is 1 in 700,000. But so many people are terrified of lightning, right? And some people do get struck by lightning, okay? Yet we scientists think that 1 in 20 is highly unlikely to happen by chance, okay? That goes to you. Go figure, okay? So, to close, it'll take a village to really bring about a change, and I hope you will join this effort. And if you want more information, you can see our links here, and thank you for your attention.
Okay, discussion. Adrian.
Thanks. I really enjoyed. And I'm well aware that P005 is no guarantee of perfection. But my question is, if you have a large effect, and that's the last thing you mentioned, and it's a positive effect, survival or death, why do you care about the power calculation that was done to select the sample size?
So that's an excellent point. And the reason is that your chances of finding a significant effect are greater the smaller the sample size.
Yeah, but the statistics is telling you that, because the p-value you arrive at is a function of the sample size, right? So.
Well, yeah, okay. If we have time, I'll come back to that, because I can say quite a bit about that. But let's see if there are other questions, and then I'll come back to that, Adrian. Okay? Anyone else? Yeah. Jim wants to ask a question.
This is a question based on my lack of experience with animal research. But in clinical trials, particularly phase 3 style trials funded by anybody, including NIH, you must include something called a statistical analysis plan in the application. And in that part, you need to say what you expect, what's the effect size you want to detect, what's your type 1 error, and what's your power. Is that typically done in the applications for animal research?
Absolutely not. Okay? That was a shorter answer than the question. Huh?
Well, should it be? What do you think?
Well, should it be? That's part of what we'll talk with Adrian in a moment when we have time, okay?
So,
Yeah,
my question is that NIH applications ask for equal number of males and females in the experiments.
Yes.
And we know that even there is a difference in the female based on the esther cycle and all. And then when we design an experiment including equal number of males and female, almost doubles the cost of the experiment, and that reduces the interest. So how, NIH can say that, okay, use the mice of this one sex, and because these are very expensive experiments, and then repeating them in both could increase a lot of work and expense.
So I get that question every single time, and I'll tell you what I think. So first of all, if you're studying prostate cancer, you don't have to look at the females, okay? So that's an advantage, or breast cancer for that matter, right? But there's
males can get breast cancer, but it's very rare.
Yeah, but not as frequently, right? And so if you look at the guidance that came out, and there's guidance that shows you don't have to double the number of animals, okay? It's less than doubling them. And they have calculations and so on that, I don't know if they go right or wrong, that I'm not going to go there. But NIH has guidance on how you calculate, how many you need in order to get a sense. And really, a lot of times, it's more about, if I were like looking at this, I'd say, for various reasons, I'm going to look only at females or only at males, but I'm going to also look at like 4 of the others. And if I see a significant difference, then I'll go back and study that more carefully, okay? So that's kind of one way around it. Now, another thing I'll say is effort and time are incredibly important things. Cost is not, because in the grant you can ask for whatever you want, okay? And if it's justified, Adrian's rolling his eyes. If it's justified, reviewers will accept it. In fact, they don't even look at budget till they finish scoring the grant. Okay. Yeah, I think also Kelly had a question here. Kelly? Yeah, I saw Kelly had a question. Yeah. Your name is Kelly, right?
Yeah.
Okay. Did you
I
forget?
thought you said Carrie.
No, I
That's why
said
was gonna
Kelly.
correct
Yeah. Trying to correct me, huh?
I just want to thank you. I really appreciate the way that you laid this out. And when it comes to the 4 pillars that NINDS implemented, it's annoying. Like, my initial response is like, why are they being difficult? And when you present it in this way, it becomes clear that they're actually pioneering, and I think that's really great. So, thank you.
Thank you.
So I want to ask what your opinion on this tension is, because
What
a lot
tension?
of I'm getting there.
Okay.
A lot of what you're saying for rigor is about group size and blinding and making sure that you're accounting for these biological variables, right? And then you submit your grant and you have all of your reasoning laid out. And then earlier we heard about the 3 R's, right? And you're getting people saying you're using too many animals. This isn't justified. How would you suggest navigating this?
So first of all, the ones who say using too many animals don't know what they're talking about.
Yeah, but
Okay.
I can't help that when they're scoring me.
Well, they're your peers, first of all, okay? But there's, in the UK, there's an institute called 3 R's, okay? And they promoted, it was initially government, and they promoted kind of the 3 R's. And when they said reduce, people interpret that as, basically do underpowered studies, okay? They don't say that anymore, okay? They now talk about, when they say reduce, is to come up with alternatives to doing the animal studies. For example, in vitro studies or things like that, okay? And because I for years told them, it shouldn't be 3 R's. It should be, 2 R's and an O. Because you need to optimize the number of animals, not to reduce them, okay? You don't want to use too many because that's unethical. You don't want to use too few because that's unethical also. So you have to optimize how many animals you use. In reality, in review, they don't really get into it in most cases. Unfortunately, yeah. But yeah, if there are no questions, I want to come back to Adrian's point.
I have a question here.
Yes, sorry. Yeah. Yes, Jan.
Yes, so I think rigors is very important, right? Like, for example, like in my lab, I can enforce these rigors like on my people. Some people, they only want to do male. I said, you have to do male and female.
Yeah.
Sometimes I said, you have to do this blinding. But in the overall regulation, like in the FDA domain, they said FDA, they have very strong guidance, right? We have to follow this guidance, like how to do the randomization, how to do this and that. But in the basic research, we don't have this strong guidance from the regulation body, right? So how do you view this point? Do you think in the basic research domain, we should also like this strong guidance, like from FDA, for example, from NIH to regarding certain practice we do in the basic research? So
I don't think so. I think what we need is to change hearts and minds, okay, and not regulation. I mean, if I hope I got across why we need to blind, and then you don't have to be told to blind because you say, I mean, I firmly believe that no one gets up in the morning and says, how can I do bad science? Okay? I mean, we want to do the best we can. And if you realize the importance of blinding, if you can, okay, then you'll do it. Okay? Now, sometimes you can't do it. I'm not saying you always can. But you'll do your best, okay? And so I think it's more about changing how the community looks at things. And it's incredibly difficult because of the perverse incentives I talked about. It's incredibly difficult. And it's not going to happen overnight. And it's interesting you say you can tell your students what to do. What I experience is the students try to tell the PI what to do, and they told that's not how we do things in the lab, okay? So it's actually, the problem is in reverse. And that's why I really want to, get to the young people. because I believe that the solution might come from the bottom up, and that's why you should listen to Damien Santola's talk. Anyone else? Yeah.
Just a quick question and sort of a comment. I really appreciate all of the points that you've been making as well. I think this is a great thought exercise for all of us to take advantage of and internalize. But one question comes to mind has to do with cost. So the purchase power of an NIH dollar is maybe about half of what it was 25 years ago.
Right.
So what's your advice on how to, pack in a highly impactful set of aims, fit in the rigor and reproducibility, and still manage the costs?
So as I said, and I firmly believe it, because I can tell you, I've sat on zillions of study sections, listened to zillions of talks, okay? And I also see what makes it to the pay line, at INDS. It's not about the cost. And in fact, the cost of grants at NINDS has gone up dramatically, okay? We're seeing fewer and fewer modular grants, and non-modular grants is just skyrocketing, okay? Because you've got to do what you got to do, Now, you also, it's not only the cost, it's what you can do. If you're proposing, things that are not realistic to do in 4 or 5 years, you'll be dinged for that. And it also wouldn't be realistic. You'll say, I need, $1.5 million to do this. It's not going to happen, okay? But in reason, the cost that you really need, I would say, put it in. And go for an unmodular grant, because these modular grants, first of all, there's no really advantage. Except that you have to itemize. That's the only disadvantage, okay? But there's no advantage in review. You sit there, there's no advantage to come in with a modular grant. No one says, oh, this is a modular grant, it'll get a better score than a non-modular grant. You don't see bias based on budget? No. I see discussions where they might say, this grant, the amount of money they're asking for is not justified in application. Okay, that I see. Okay,
So
but I don't see that they're saying, "Oh, this is expensive. Let's give a better score to something that's not that expensive." Okay.
So, Shai?
Yes.
Over here.
Yes.
So, I clearly the goal is aspirational and very needed, which is to clearly generate data that's reproducible, right? That's the goal. So, but I'm going to kind of ask the question that's kind of a takeoff of some of the questions here with respect to cost and issues of that nature. But it really is, it seems to me that some of the things, it's kind of interesting. We were actually at lunch just by chance. I was complaining about a particular paper that we were publishing, and
you were complaining?
all the things you had to, the hoops you have to jump through, and Adrian was talking about a paper that he's currently going through. So a lot of this has to do with these new rules, right, with respect to dumping all of this information and data and everything into these into the formatting process. And I guess my point is the following, which is that seeing a problem is very important. I think finding reasonable and good potential solutions is also very important to actually make this happen. So the process of simply saying, well, this is going to take lots and lots of years is one way of addressing it, which to me is just kicking the can down the road, right? Mm-hmm. So when I think that, and I don't know, there may be initiatives and discussions about this already, but you need to have reasonable solutions for investigators rather than telling them, well, you need to have 20 animals or 40 animals per group, which their budget cannot take on, or you need to have X, Y, and Z, which is perhaps not something doable, or in this case, dumping all this information, which we know that the next investigator is not going to look at. They're not going to look at individual statistics of every animal that's in your study and looking at all the different parameters associated with it. It's very, very difficult, and simply frankly not realistic. So I think that's the, you know, the question is, here's the problem, but start to develop at least an outline of the solutions that you need to have for them.
Okay. So first of all, I'd say that I think a month in Australia didn't do you any good. And the other thing I'll say is you can look at my shoes. It doesn't look like I kick any cans down the road, okay? I've always said, and I continue to say, never ask what someone else can do. Ask what you can do, okay? And that's my philosophy. And that's where I am, where I am, okay? I didn't say, the journals should deal with it. No, I brought the journals in and we had a conversation, we had a discussion, and we got change made, okay? Got NIH to make change. It's always about what I can do, okay? Now, things are expensive. I'm not saying they're not, right? And I always say again, I say something which is no one wants to hear, okay? Do less, but do it properly, okay? What's the point of doing an underpowered study unless it's exploratory, you're just looking for things. I'm not talking about that. I'm talking about when you're doing confirmatory studies. What's the point of doing that underpowered, okay? Don't do that experiment. Either do it properly or don't do it. So you should ask for what you have, what you really need, and plan what you can really accomplish properly. That's my philosophy, okay, of how I do science. So you really kind of led right into the conversation with Adrian, and what I want to say is I'm going to take an hour talk that I give and summarize it in 2 minutes, okay, which is not going to be easy. How did p-value come about? So there's Ronald Fisher, who came up with the t-test, okay? And in his view, you have a single hypothesis. It's the null hypothesis. It's just one hypothesis. And you ask how unusual the results are. How, do they look what he called significantly different, but what really meant was, are they in harmony with the null hypothesis, or should we be looking at them more carefully? Not, do more experiments. That's what he meant, okay? The community was not happy with that, because now what you say, all he says is, I have one hypothesis, the null hypothesis, and look, these results look different. So, now what? So, Neiman and Pearson came into the picture and said, let's have an alternative hypothesis, okay? So now we have two hypotheses. Now, what Fisher said, and this is important, assuming the null hypothesis is correct, okay, what is the probability that we'll find results that are equal to this value or greater, okay? That's all he said, assuming the null hypothesis is correct. Neiman and Pearson came in and said, assuming the alternative hypothesis is correct, okay, what is the probability of detecting the alternative hypothesis or having an error, which we call the type 2 error, that will miss it, okay? You cannot have both true in the same experiment. You cannot have both hypotheses true in the same experiment. And both Fisher and Neiman and Pearson never said that a single experiment is the solution. They said if you do it many, many, many times, on average, you'll be correct, this amount of times or that amount of times, okay? That's all they said. Now, how can you publish in Nature if you can't say about a single experiment that you found something, okay? That's unsatisfactory. So what did the community do? They decided to ignore the sample size calculation, or the power, calculating power, okay? To ignore the effect size, they don't actually estimate effect size, and decided to worship the p-value, okay? Now, it's totally absurd, okay? And every paper that you read, for decades of serious statisticians say, it's absurd. There's no statistical justification for what we do today, that we worship the p-value, okay? But that's where we are, okay? And that's the other talk that I do about increasing awareness, that what we're doing is totally ridiculous. And, you know, and you have to be more humble and not to say, you know, we discovered something. You know, you can say, this suggests that, or this might be, or this is worth looking further into. You cannot claim a discovery based on p-values, okay? So that's kind of where I come in. So just to make that point, okay, I can think of 2 studies that looked in neuroscience on sample size calculation and power, okay? The average power in one of the studies was around 20%, okay? 20%, okay? The other study, it ranged from, I think, 18 to 30% or something like that, in that range. The second study said that 50% of studies in neuroscience had a power of 20% or less, okay? And that shows that actually we're ignoring sample size calculation and power calculation, okay? So we have a long way to go to improve things, and we'll see where it heads. Yeah.
Okay. Well, I think that's terrific food for thought. And I honestly found that to be one of the most lucid discussions of the problems with the p-value. But it is definitely one that you can read a lot about. And actually, lots of debates about from statisticians themselves on how to think about the p-value. And actually, in our institution, our statisticians are moving away from p-values and going to confidence intervals and ratios and giving you kind of a sense of how likely the events are due to chance or not to chance.
I will summarize by saying if you take 2 statisticians and put them in the room, you get 3 opinions. And that's really what's happening in the field. There are so many different opinions, you know. And my disclaimer in my other talk is that I'm not a statistician, I'm not a historian. But I've read hundreds of papers, you know, going back to the 17th century about statistics, okay? And it's incredibly complicated, okay? It's not straightforward, but what we do now is ridiculous. Yeah?
Okay.